This is the last lecture, lecture 25, the genetics of complex disease. So in the last lecture before this, we discussed quantitative genetics, and human disease is often actually characterized more as a function of many, many different genes which contribute to complex disease. So obviously there are some cases where the disease, the genetic basis of the disease is not complex, so cystic fibrosis, Huntington's disease, but most diseases that we have to face in our lifetimes are caused by many, many different genes. All right, so these are called polygenic diseases, and they're complicated, and we want to understand what their causes are. So examples of complex human diseases, as opposed to cystic fibrosis and Huntington's disease, are heart disease, Alzheimer's, cancer, depression, diabetes, Crohn's disease, irritable bowel syndrome, lupus, asthma, go on and on and on and on. And all of these diseases are due to a combination of genes and the environment. But as geneticists, we'd like to identify the genes that contribute to the increased risk of getting these diseases. So to explore how, as geneticists, we try to uncover the genetic basis for complex diseases, I'm going to use two different examples. I'm going to talk about autism and type 2 diabetes. So we're going to start off with autism. So to understand the genetics of autism, it's commonly performed population studies to identify whether or not there is a strong genetic basis to autism. And it turns out that siblings of autistic children have about a 25-fold greater risk of also being affected. So even, not identical twins, but dizygotic siblings have a 25-fold greater chance of being affected, which suggests that there's a genetic component. And if one identical twin has autism, there's between a 60 to 90% chance that the other one will as well. Again, this indicates a significant genetic component. And for fraternal twins, or dizygotic twins, the concordance rate, the rate at which one individual has it and then the other one does as well, is 10%. So all of this work has revealed that there's probably a broad sense heritability for autism to be about 0.7. But there are some complexities associated with this. First off, autism has many different causes, right? And it's sort of not necessarily a single phenotype. So, for example, cancer is a catch-all description of the consequences of varying conditions. So, there may be very different reasons why two different people might have autism, both genetically and perhaps environmentally. Autism can be caused by rare new mutations, as well as low-frequency old mutations that segregate in the population. And there's an interesting observation that males have a much higher risk, suggesting that there might be some sort of interactions between the genes that cause autism and the sex of an individual. Now, type 2 diabetes has a different pattern of genetic contribution. And just as a reminder, what type 2 diabetes is, there's two types of diabetes, both of which are involved in the inability to regulate blood sugar levels. Type 1 diabetes is caused by an immune response that attacks cells of the pancreas, which is the source of insulin. So that means that insulin is not properly regulated in the bloodstream of an individual that has type 1 diabetes. Insulin, of course, is very important for regulating blood sugar levels to allow a person to, uptake glucose from the bloodstream or release glucose into the bloodstream. So it regulates the levels of glucose in the bloodstream. And type 2 diabetes, however, is different. All right, it's not necessarily caused by a genetic basis where there's an early immune response. There seems to be a contribution of the environment. So individuals that have type 2 diabetes are unable to regulate their blood sugar levels, which can have very big consequences. But the reason for it is not a simple sort of early event that happens in life. It's a complex event that typically happens later in life. So the spectrum of type 2 diabetes varies widely among different ethnic groups. So it's frequently about 2% among European Caucasians, but there are certain native groups that have like much higher incidence. So for example, in the Pima Indians in Arizona, it's about 50% prevalence. So there seems to be some genetic component. So for example, there's a fourfold greater risk with an affected sibling. But one thing that's important to think about with this type of interpretation is that siblings will frequently share the same environment. So is that fourfold greater risk due to genetics, or is it due to the shared environment? Now, some genes have been identified that seem to influence the predisposition towards type 2 diabetes. And there is one gene that's been found called TCF72, and disease increases 2.4-fold when homozygous for the allele that is associated with type 2 diabetes. But overall heritability seems to be low, about 26%, which you can see contrasts significantly with autism. So the complexities of type 2 diabetes, there's a very strong environmental component. Another thing to note is that the increase of type 2 diabetes in the world's populations is significant across many different countries. And so this has been proposed to suggest that there's a very strong environmental component, and particularly the dominant environmental component that's probably contributing to the increase in type 2 diabetes is a change in human lifestyle that's associated with increased obesity. So we have these two different diseases. We have autism and we have type 2 diabetes, and they're basically have different genetic contributions. And as a geneticist, what we'd like to do is identify all the genes that contribute to broad sense heritability for these complex traits. So if heritability is estimated for about 70% for autism, we would like to identify all the genes that account for that. And if heritability is estimated to be about 30% for type 2 diabetes, we'd like to identify all the genes that account for that. How do we do that? Okay, well, there are two approaches. Now, the classic approach for identifying the genetic basis for disease, which we have already discussed, is to use families. So over on the left here, we have a family approach in which alleles are linked to the phenotype in pedigrees. All right, so for example, Huntington's disease was identified, the genetic basis for it was identified through a very large pedigree in which, there was clear transmission of an allele through the pedigree across multiple generations that caused Huntington's disease. And so a standard Mendelian approach can be used there, where one can use linkage analysis within families to identify regions of the genome that are associated with an increased risk of a disease and therefore likely indicate where the gene is that is the genetic basis for the disease. Now, that's tricky, however, with complex traits, because one can't easily sort of look at a family and just say, oh, yes, no, yes, no, yes, no. There's not a Mendelian property, because there's sort of like not enough individuals within a single family to get a significant amount of resolution. So another approach to identifying the genetic basis of complex traits is to use unrelated individuals, a very large sample of unrelated individuals, and just basically ask which alleles simply show a correlation in the population. And this gives us the definition of a genome-wide association study, which is a tool for identifying the genetic basis of complex traits. And I'm going to show you now how a genome-wide association study can identify the genetic basis of complex traits. So the way that this is done is within very large samples of populations, one can collect individuals that are affected by, say, disease, heart disease or cancer risk or type 2 diabetes. And one can take that and then match it. And you try to keep the environments about the same because you don't want to be confounded by the environment. So you collect cases and you collect controls, individuals that don't have the trait. And what you do is you genotype everyone at thousands of markers. So this is with just one marker, and it would be SNPs, right? Remember we talked about the SNP array, that you can take the DNA from an individual, you can put it on a SNP array, and you can determine the genotypes of many, many, many single nucleotide polymorphisms across the entire genome. At this position, do you have a G homozygote or a GT heterozygote or a TT homozygote? Okay, at this other position, do you have AA or AC or CC? At this position, you just go through the entire genome and genotype everyone at thousands and thousands of markers. And what you look for are allelic variants that seem to be associated with the disease. And so here we have, and this is for a smaller number, we have our cases, right, which is a couple hundred, and we have our matched controls, which is a few hundred as well. And what you can see here, in terms of the frequency of the big A genotype, and this is any particular random location in the genome, it's not one that you know to be associated with the disease. The abundance of this genotype, there's 65 individuals that are big A, compared to, in the control, 16 individuals that are big A. And then if you look and you compare the incidence of big A, little a, maybe it's 133 or 81, and here it's 202 versus 316 for little a. All right, and one can see that if you compare the total numbers, there seems to be a much greater increased incidence of the big A genotype when in the individuals that have the cases of the disease versus the individuals that don't. And one can do a statistical analysis of these associations, either through a chi-square test or something called the Fisher's exact test, and show statistically, in this case, that the A allele, the big A allele, seems to be more common in individuals with that disease. And that suggests that you may have found a gene that contributes to variation in the disease trait. One thing that's important to remember with these types of studies is those variants that are identified aren't necessarily what are called causal, right? They may not be a variant that actually causes the disease. They simply may be physically linked to variations that cause the disease. So for example, in a big SNP genotyping assay, so this is DNA sequence along a region of the genome, one might find, for example, that a SNP here having a G a nucleotide as opposed to an A might lead to increased risk, say for example, of heart disease. It doesn't mean that this G nucleotide causes heart disease. What it means is that there's something about individuals that have that G nucleotide that have an increased risk of heart disease. And if one looks further down, one could see that the causative mutation may be somewhere else. So here we have individuals G, 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 and A on top. Okay, and all of these individuals that have G, 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 G also have C, 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 C. So statistically, one might see an increased association of the G allele with heart disease, but it might in fact be the C allele, which may not be in the, in fact, SNP genotyping array, right? It may be the C allele that's causing the increased heart disease. This allele may be in our genotyping array. This allele may not be. But this still allows us to basically narrow down into the physical positions of the genome where the genes might lie and where the variation may lie that contributes to increased disease risk. Okay, so we're going to walk through a couple genome-wide association studies. One that was done with type 2 diabetes. This is a paper that came out a few years ago. A genome-wide association study identifies novel risk loci for type 2 diabetes. In this experiment, there are 1,363 French individuals included in the study, split between individuals that have type 2 diabetes and then controls that didn't. And 392,935 SNPs were genotyped. So this was the single nucleotide polymorphisms that are in the genotyping array. So each individual's DNA is taken out of their, collected, and then determined for a given individual at this position, let's say position 1, are you AA or AT or TT? And at position 2, are you TT or TG or GG? So on and so forth. And these are distributed throughout the entire genome. And what does one find? Okay, what I show on the y-axis here is the fold increased risk associated with different variants at different regions of the genome. And so in red, all right, are ones that are statistically significant using this statistical approach that I described earlier, all right, either using a chi-square test or a Fisher's exact test. So these are ones that are statistically significant, and then one can see the increased risk. And what in each of these little plots, 1, 2, 3, so on and so forth, I don't show the entire genome. Each one of these indicates a chromosome. So this is chromosome 1, chromosome 2, chromosome 3. This little dip in the middle there represents the location of the centromere, which is hard to genotype because there's a lot of low complexity sequence in there. And each of these dots represents a given SNP, a given single nucleotide polymorphism in the array, evaluated for whether or not there's a statistical association with increased risk of type 2 diabetes. And one can see that along chromosome 1, there's 1, 2, 3, 4, 5, 6, 7, 8. 8 regions of chromosome 1 that seem to have a higher abundance in individuals that have type 2 diabetes. Chromosome 7 doesn't show any. Chromosome 13 shows that one there. And one can see that these are all candidates now for places in the genome. This is an interesting one because they all seem stacked right on top of each other, so that might be a really strong one. But these are all candidates for regions of the genome that might have genes that have variants in those genes that increase or decrease the chance of type 2 diabetes. And then, using a different population, it was confirmed, 4 of them were confirmed, right? So these other ones using a different population didn't pan out, but this one, this strong one I mentioned earlier, and this one here were all confirmed to replicate, all right, in a different sample, and they altogether contribute to 70% of risk. So what's important to think about now, though, is will that be the same loci identified in the same groups, right? So both of these populations that were used to confirm were European populations. But it doesn't necessarily mean that the genes that cause, the alleles that cause risk of type 2 diabetes in, say, for example, France, they may be very different in different other parts of the world. For example, the alleles that the Pima Indians have that increase the chance of diabetes might be completely different alleles. So that's important to think about with these types of studies. And the most important thing to think about is are your samples diverse across the world? Or are your population samples biased? Here, of course, our population is biased towards France. Okay, here's another type of genome-wide association study that was done. This was done for autism, titled "A Genome-Wide Linkage and Association Scan Reveals Novel Loci for Autism." And so in this analysis, 1,553 affected individuals were genotyped in addition to controls, and they were genotyped for 500,000 single nucleotide polymorphisms across the genome. 8 candidate regions were found, but only one of those could be replicated in a second data set. In addition, this was disheartening. Previous studies had identified candidate genes associated with autism, but in this large study, those identified candidates were not confirmed. So this is complicated because in the diabetes case, they were able to validate those genes in another population. Here, in the attempt to replicate across different studies, they didn't match up, the same genes didn't match up across these different studies. Why is that? And in this paper, there's an important point that was made. They stated, both our linkage and association analysis suggests that low frequency okay, less than 0.05 frequency, minor alleles may be common in autism. Let me just restate what they're saying there. What they're saying there is that the alleles that cause autism might be at very low frequency in the population. And there may be many of them. It turns out those may be very hard to find. And let me explain why they are hard to find. Low frequency alleles are commonly alleles that may have been caused by recent mutation. So, for example, if you go 2 generations back, right, to your grandparents' generation, there may be new alleles arising either from your grandparents or your parents, perhaps, that lead to increased risk of autism. But that increased risk of autism, which would show up as a family association, those allelic variants will be different in different groups of families. So you have, many different families. There could be an increased risk of autism within those families. But the alleles in, say, family 1 that cause autism might be different than the alleles that cause autism in family 2 or family 3 or family 4. And what genome-wide association studies do is identify common variants that are associated with the disease risk. So if the variants are extremely low abundance, it's going to be hard to identify them. You can think about this by asking, how would a GWAS, a genome-wide association study, find new variants that are constantly arising each generation? And it's going to be very difficult to do. So what this leads to is some challenges for genome-wide association studies. There's this question about why sometimes there's a failure of replication, whether or not a population sample in one case is applicable to another population. There's something called the missing heritability. Even though we've known for a long time that height has a strong genetic component, many genome-wide association studies have been done that have not been able to identify all the variation genetically that's associated with height. Although more recently there have been better cases of studies that have been more successful in finding that. And there's a whole host of statistical problems. So there are a lot of challenges with genome-wide association studies, but they do hold a promise to identify the genetic basis for complex diseases.